Hi, Basu, and today I'm going to be telling you about genomic tools that have come online recently being used in biology and for the treatment of human disease. I am an assistant professor at the genetic medicine section in the Department of Medicine in University of Chicago, and also a staff scientist at Argonne National Lab in the Center for Nanoscale Materials. The idea is to build new devices, new imaging, and new materials, and bring them to medicine, both for genetics and in general. So I'm going to start with gene expression in complex tissues. And I'm going to point out that we have the same DNA in our whole body, but we perform many, many different functions. We have many different cell types. For example, in the brain, we have 100 billion cells and at least 1,000 different neuronal cells that does the different function. And that is just for the brain. For the rest of the body, there is an enormous complexity in cell types and functions that exist. How does this happen? It happens through gene expression. So with the same set of DNA in all the cells, we have different parts of the DNA expressed, which is expressed in the form of RNA, messenger RNA, and they go on to produce proteins, which are the actual functional part of the cells. And that is how the cell performs its function or displays its phenotype. So we are going to classify different cell types by gene expression. So that can be done in many different ways by looking at RNA, for example. It is easy because RNA is expressed to a larger extent, many more copies of the RNA than the 2 copies of DNA that is present. You could also be looking at the proteins in a given cell. Or you could be looking at the parts of the DNA that are accessible for transcription or for gene expression. We are going to be talking mainly about messenger RNA or RNA. We are going to be talking not just about profiling single cell for the gene expression, but we are also going to talk about throughput. It is not enough to look at single cells, but it is very important to look at many cells, thousands of cells, tens of thousands of cells, if we are to understand the complex nature of these tissues, like the brain, the immune system, complex tumor microenvironment, for example. And in the very beginning, around about 2013-14, when single cell experiments were becoming a norm and the field was adopting it, one of the challenges in scaling this kind of single cell sequencing technique was throughput. How could we perform many cells in parallel, and how could we reduce cost? Now, we are all aware of the high cost of sequencing, DNA sequencing, and there is also in parallel a very high cost of preparing these cells ready for the kind of sequencing that the field does these days. So we had to perform many experiments at the same time and also be cognizant of the cost. And for that, the technique that got very popular was emulsion microfluidics. These are little droplets of water in oil, where these water droplets serve as reaction compartments. So you can imagine each water droplet as a test tube, an individual test tube. And if you were to do a 10,000 cell experiment, you have to generate 10,000 such compartments, 10,000 such water droplets. This was done by using microfluidic devices and using water and oil emulsion. I'm going to be referring to this as droplet microfluidics. The other thing that we needed to do was to do a to assign labels to each of these water droplets or reaction compartments. So if we had a cell in each of these cells, in each of these droplets, there was a necessity to attach droplets or two of the cell in a given droplet. And this is traditionally done or mostly done with DNA barcoding, though there are other options of doing this. For example, fluorescent barcoding. We are going to be using a fluorocarbon oil, which is heavier than water. It's very low viscosity. It is also pretty compatible with cells, with living cells. And we add a surfactant to these droplets or to these water oil emulsion system so that we can have these droplets be stable for long periods of time. So what we have here is a microfluidic device-generated water and oil system. The picture to the very right here, water droplets getting generated in an oil background. These are stabilized using a surfactant, which goes through the interface of the water and the oil droplets and stabilizes it for not only long periods of time. You can have these droplets be stable for months at room temperature, but is also stable under thermocycling. So if you were to do PCR, for example, it is possible to do thermocycling and still have the droplets maintain their integrity. It is possible to grow cells in them. So if you were to load these cells and do incubation, you could also do that. So it is not just useful for doing single cell genomic applications, but this reverse emulsion system is good for a variety of other purposes also, which I'm not going to go into, but is a field in itself. And to mention the DNA barcoding that I had talked about earlier that is necessary to label each of these cells, we assign a DNA RNA that we get from these cells. So for example, if we are interested in the messenger RNA, we are going to attach this DNA barcodes to the messenger RNA. If we were to look at a particular DNA fragment from the cell, we would attach this DNA barcode to that DNA fragment. The unique thing about this DNA barcode is that it has a series of J's here. If you look to the bottom of the slide in blue, this is going to be unique for every droplet. So each cell that is assigned to a given droplet is going to have its own unique barcode. There is also a sequence called UMI, or Universal Molecular Identifier, that is going to be unique to each messenger RNA that lets us do RNA counting. And there is also a section of poly-Ts here that is going to select out the messenger RNA. So mammalian or eukaryotic messenger RNAs have a sequence of A's on the end of the messenger RNA molecule. The poly-T allows us to select out the messenger RNA from all the other types of RNA that is present. I should mention that the mRNA is a small fragment that is present. They are also very informative, but not for the purposes of classifying cell types. So we are going to ignore those for the most part. For the technique that I am going to be talking about mainly, this DNA barcode is attached to a polystyrene bead, actually a PMMA bead, that is 40 microns in size. And there are many such DNA barcodes on the surface, attached to the surface of this bead with peg links. And by our estimation, it is roughly 500 million of these barcodes, DNA barcodes that are present. We have many such beads. Each bead is going to have a unique cellular barcode. That's the part in blue. And each one of them is going to have a high complexity of UMIs, about 500 million of such UMIs, roughly speaking. That is going to allow us to capture all the RNA that's present in mammalian cells. So why droplet microfluidics? There are other ways of doing these kind of techniques also, such as microwells or nanowells. People still do a lot of plate-based single-cell sequencing. A regular plastic microwell plate, for example, has 96 wells where you can easily process 96 cells. What this droplet microfluidic allows us is to just scale up this number many folds. Not only does it allow us to scale up the number of cells we can process at a given time, but we can also use very, very little reagents. Now, people who work in biology or in genetics, we know how costly enzymes are. This kind of scaling down of the reaction compartments from 10 microliters to say 50 nanoliters allows us to save on reagent cost, which is important because we are doing 10,000 such reactions, and these numbers quickly add up. Other advantages of microfluidic devices are that the channel sizes, the dimensions associated with the fluid flow is very small. And we have very low Reynolds number flow. The droplet generation, the reactions that happen are very happen in a laminar fashion, and there is a lot of precision and reproducibility in these droplets generated. The other advantage is that these droplets get generated at very, very high speed. You can go anywhere between kilohertz to up to 20 kilohertz. That's 20,000 droplets getting generated per second. So it really helps with like reducing batch effects, for example. And of course, you only have to run a longer experiment to change the number of cells that you need to process, which is a distinct advantage. So the first technique that I'm going to talk to you about is Roxy, which we use in the lab quite a bit. It uses this emulsion microfluidics and DNA barcoding technique that I just introduced. You start off with a tissue of choice, complex tissue. Let's think about the brain, for example. You start with a cell dissociation. It could be mechanical, enzymatic, detergent-based. You dissociate the cells. I'll also point out that this is the most critical part of the entire process. You start with the cell dissociation, generate a single cell suspension such that there isn't a lot of leaks in the cell membrane. You don't damage too much during the dissociation process. If the cell membranes get leaky and damaged, there will be some amount of RNA that will leak out into the ambient solution, and that is going to add to the noise. So we have to be cognizant about that. But we also have to make a good enough single cell suspension such that they are all separated and cells don't stick to each other. After this, we are going to flow this into a microfluidic device with a DNA barcode bead that I had talked to you earlier. We are going to flow them in such a fashion that there is one cell and one barcode bead in a given droplet. This is mainly done by adjusting the concentration of the cells and the barcode bead. Once the cells are encapsulated into, there is also a lysis agent that was flown in with the DNA barcode beads that is going to destabilize the cell membrane and have the RNA released into the droplet. And what is going to happen is this RNA, because of the high concentration of molecules in a droplet, it's going to find DNA barcodes that are attached to the beads and come to the poly T section that I had shown you before. So the end result is we are going to sample the mRNA from a given cell onto using these DNA barcode beads, such that a fraction of the mRNA is hybridized onto the bead. Once this happens, we can then break the droplets. We can then perform reverse transcription. This can be done both inside the droplets or outside the droplets. And there are different techniques that do either or. Once the reverse transcription happens, the RNA gets converted into stable cDNA, which are complementary DNA that has the barcode, the DNA barcode attached to it. We then collect all this DNA barcode and DNA barcoded DNA and do amplify and make library or sequence. For most of the cases, Illumina-based sequencing, and that is a standard that is practiced in most cases. So here we have a Dropseek device setup that is run by syringe pumps. There is the device sitting. The droplets are getting generated in the microfluidic devices where the cell beads and lysis agents meet. The cells are getting encapsulated in these droplets with the beads, and then they flow out. They are generated at 2,000 droplets per second. And this is, for your information, this is a 125 micron device that generates about 125 micron droplet in diameter. A couple of things that I'm going to point out about this technique is that it has pretty high correlation with bulk RNA-seq, about 0.9. On average, we get about 5,000 unique genes for every cell that is run. Now, this number is going to change by the cell quality genes that are being expressed, that are getting cycled, and that roughly translates to about 12 to 15% of the RNA that's present in a given cell. So not all the cell, we are only capturing a small fraction of it. That gives a representative picture of the cell. To establish that we can indeed do single cell library and it's not a mixture from different cells, we perform an experiment taking mouse cell lines, cells from a mouse cell line and cells from a human cell line, mix them together, run Dropseek. And for the most part, after sequencing, when we align each cellular barcode to the mouse and the human genome, we see that all of them align to the human genome or the mouse genome specifically, mostly. There is a small scattering of dots in the center. I'm looking at the panel to the bottom left. There's a small scattering of drops or cells that is that have transcripts from both the mouse and the human genome, and that can be droplet, both a human and a mouse cell got encapsulated and processed together. So it shares the same barcode. The number of such cells would be double that. So if, let's say, this kind of scatters are about 2% of the total number of cells, our number of doublets or that have 2 cells would be 4%, for example. Another thing to point out in a Poisson loading framework, in that we load the cell concentration and the bead concentration, or we adjust them in such a way that there is only one, the probability of getting one cell and one bead is pretty high. That means a lot of these droplets are empty, and a lot of these droplets have one cell and no bead, or the other way around. Now, we can, depending on these loading concentration, you are going to have doublet rates change. So, for example, we talked about a 2% or 4% doublet rate in the previous slide. You can change the loading concentration. With a higher cell loading concentration, you're going to get a higher fraction of drop. It's going to be a shorter, quick and dirty experiment. You can lower this cell loading concentration. You can get a clear, a more cleaner experiment, and this is done by choice, by what your experiment requires, for example. So we see in these different panels, by changing the cell concentration, the loading cell concentration, you can decrease the number of dots that are present. And it is not only depend on the loading concentration. That would be the most ideal situation. If you have cells that are leaky, for example, that's going to give you ambient noise that could give you more even mixing of these scattered plot scatters in the species mixing plot. Or if you have cells that are sticky, then you might have a higher chance of getting a human cell stuck to a mouse cell, and you can get a higher number, for example. What are these kind of experiments useful for? They are good for looking at complex tissues. By looking at their RNA expression, we applied this technique to mouse retina for very young mice. And we see that for the most part, the bioinformatic analysis yields us these cell types that can be matched to what is already known for the mouse retina. So, for example, these blobs actually are color-coded by the picture to the left. So this kind of plotting is called a TSNE plot. There are other ways of doing this. Principal component is one way that is going to show you the principal components 1 and 2, 2 and 3. TSNE is a higher dimensional representation. It shows you many more components. It captures many more components. There are commonly used type called UMAP. But what it shows you is this high dimensional data. So this is about 10,000 cells with 20,000 genes, potentially 20,000 genes expressed. So this is a pretty high dimension data. You can show this or represent this in 2 dimension. And the cells that are closer together, they cluster by similarity. So all the cells, let's say in this navy blue blob, they are going to have very similar properties and are going to be that much different from the purple blob, for example. And in our initial experiments, we were able to run 50,000 cells approximately and get about 40 different cell types. So that was a pretty big jump in the number of cells we could process and also the number of different cell types that we could capture in an experiment. Now, one of the challenges in using Dropseek or any kind of single cell RNA-seq is that we are going to have this problem of cell type dissociation where the quality of the experiment depends on the cell health or the dissociation quality. Now, if we were to work with frozen tissues or postmortem tissues, the cell quality is already pretty low, and that leads to a lot of RNA leakage and RNA degradation. And that is not very conducive for the Dropseek protocol that I had talked to you before. So I'm going to talk about a very, very similar complementary technique called droplet single-nucleus RNA-seq or Dropseek that allows us to work with a little bit harder to work with tissues. So this was developed to look at brain tissue. We remember we looked at retina, which is not really brain tissue, but we were very interested in looking at the complex cell types in the brain. The way to do that was to look at the nuclear RNA. And we looked at nuclear RNA because the cytoplasmic RNA was not accessible to us. We couldn't generate a good enough single cell suspension because all the neurons are very well clustered or tied to each other by the axons and the dendrites. So it's very difficult to make a clean single cell suspension. So the choice was to work with the single nuclei. The nuclei is much more intact as RNA. It is the RNA that got recently transcribed and is in the process of going out from the nucleus to the cytoplasm. And it is similar, it is representative of the cell, but not quite the same. So in comparison to the single cells RNA, you are going to get a smaller fraction. This is roughly a tenth of what you would expect from the cytoplasm or the whole cell in general, because most of the RNA is present in the cell, in the cytoplasm. And this is a very recently transcribed RNA, so you're going to get a pretty recent snapshot of what the cell just did, or what the cell is transcribing at that particular point of time. It can also be not completely formed. It can be, it is nascent. It might be missing the polyadenylated tail. It could still have its intronic regions. So to point out, in the lower panels, in a single nuclei RNA-seq, we roughly capture half the number of RNA present in the we capture roughly half the number of RNA that we typically capture with single cell RNA-seq. And this is, the fraction is a little bit higher in comparison. Even if it had a tenth of RNA, we capture higher number of genes, and that's because of the way the protocol worked out. I'm not going to go into the detail. But if we compare the single cell RNA with the single nuclei RNA, that there is a much higher prevalence of non-coding RNA in the nuclear RNA and mitochondrial RNA from the cellular RNA or the whole cell RNA, which is expected. And if we were to look at which particular regions these RNA map to, it would be on the there would be a smaller fraction would map to the exonic region, relatively higher fraction maps to the intronic region, some intergenic regions, and so on and so forth. So this allowed us, the single nuclei allowed us to adult brain, even though we weren't able to get good single cell suspension out of brain tissue, adult brain tissue, we were able to get single nuclei. And based on the nuclear RNA signature, we could identify cell types from this protocol. And I should also point out that this tissue was obtained from the GTEx project, and it was archival tissue also. So not only for adult brain tissue, but this was good for frozen samples that were sitting for about 5-6 years in the freezer before we ran this. So the cell quality was pretty compromised to start with, and we were still able to make it work, which was very gratifying, and opened up our options for looking at many difficult to work with tissues. I should point out that if you take any one of these big blobs, so for example, in the TSNE plot to the left here, the green blob, and if we perform hierarchical clustering on these, you can find out even finer structures in a given blob and identify finer subtypes within a given cell type in the tissue. And even though we see from the plot to the bottom right, even if they have different numbers of RNA captured for a given cell type, it clusters by cell type and not by the number of RNA captured. It's also something to note that different cells do have different number of RNA expressed. We are applying single cell and single nuclei RNA-seq to a bunch of different experiments. One of them is benchmarking these two techniques using human iPSC-derived cardiomyocytes. This was done for the Human Cell Atlas, And what we did was we sampled cells from different points of the differentiation process. It was a 15-day differentiation process. By day 7, adult cardiomyocytes are already established. We take day 0 to all the way to day 15. And we samples by using both Dropseek single cell and Dropseek single nuclei. One of the things to note here again is that there is a lot of intronic reads and the single nuclei data that is not present in the Dropseek data, and that is because we are working with mature RNA in Dropseek versus pre-spliced RNA in Dropseek. And again, the number of RNA that we get from intronic regions is much higher, and we use this kind of intronic reads into our pipeline now to increase the number of RNA that we get, and that helps us improve the data quality. We did that on these cardiomyocytes, differentiating iPSCs into cardiomyocytes, and see that for both Dropseek and Dropseek, we capture the major cell types from day 0 to 15, and that there are different types of cardiomyocytes, subtle variations in cardiomyocytes that are generated by these differentiation protocol. We also see that these two techniques are pretty good in capturing the fraction of cells in each of these time points. And that was also very gratifying to see. And I'm going to point out that this was possible because these cells were growing on a plate. It is much more difficult if you were to create a primary. Lots of bias is generated tissue in itself, but at least using these two techniques, there wasn't any technical artifact that was giving different cell fractions. And using, again, sophisticated bioinformatic techniques, looking at their expression, we can construct a trajectory in which the cells differentiate, and we can not only get this, recreate the differentiation process into the major cardiomyocyte types that we see in our, both these techniques give very similar results. I should also point out that there's this very small fraction of cells that the single cell protocol it manages to identify that is not identified by the single nuclei protocol. And with that, I am going to stop my presentation.
Alrighty, awesome. We are all set, and you did a great job. That caught me off guard for a sec. I was tracking with your slides, and then you, I thought you had probably half your presentation left.
That's correct. You know, I couldn't have covered everything. I decided I wanted to tell a story, and I think this is where it stops. There is another part that, you know, I could have gone into, but there isn't enough time. Okay. I wonder if I could add the one more slide, which is the acknowledgement slide, but we can also cut the presentation here.
Yeah, if you want to go ahead and jump over to the acknowledgement slide, we can record that real quick while we're still on. Let me just make a quick note for that before we start the last slide there. 1 second.
So we were in slide 17, and now we're on 30.
Yes.
Yeah, that was like, yeah, half the talk.
Yeah. Okay, I made a note. So what I'm going to do is I'm just going to count you down from 5 real quick, so we can main cut for the acknowledgement slide, and then you can get started after I count you down. Alrighty, starting in 5, 4, 3, 2.
Last but not the least, I would like to acknowledge the people who did all this work. This is not possible to do such a huge amount of work without a big team drawing from device fabrication, molecular biology, biologists, computational scientists. And we have a very big group working on this. A lot of the work that I talked about today was done in my postdoc at Broad Institute, but a current position at Argonne and UChicago, I have my group to the names are listed to the left. This is my group outing, a picture to the right. Very good colleagues without whose help I cannot function, and some colleagues not in Chicago who also participate in other projects that relate with Dropseek and Dropseek, and of course funding agencies that keep our lab running. Thank you.
Okay, great. All set. So pretty much from here, I'll just do some quick editing on the front and back ends, and then I'll work on stitching together where you finished off on slide 17 with your acknowledgement slide, and then we'll be all ready to go for the presentation on Wednesday.
Thank you.
Of course, any other questions for me before we wrap up?
Not really, but so all the things that I said today, I talked about today up to slide 17, it's all published, so it should be okay to share.
Oh, perfect. Okay, I'll go ahead and update the note then. That's helpful. Perfect. So I'll go ahead and just use up to slide 17, and I'll make sure not to use any of those other slides.
Thank you.
Alrighty. Well, we will talk to you soon. Thank you for your time. We appreciate it.
Thanks, Tyler. It was a big help.
Have a good rest of your day.
You too. Bye-bye.